Hello, everyone. It's my pleasure to be with you at ESER to discuss what optical genome mapping offers for the study and the characterization of cancer samples. Among all biological tissues, cancer is probably the most complicated to interrogate with genome instability, the high cellular heterogeneity, and this explains the limitation to NGS to tackle this type of sample and leading to a lack of understanding of the role of structural variation in cancer as of today. The way our technology operates is not based on sequencing and can be more compared to karyotyping, but at a much higher resolution. With karyotyping on the left, chromosomes are stained in a way you can distinguish a pattern of bands that is unique throughout the genome, and any abnormality in this labeling pattern would indicate the presence of a chromosomal alteration. The same way with optical genome mapping, a pattern of labels is applied throughout the genome, but this time with one label every 5 kb on average. And this will be scanned and analyzed automatically by the Sapphire system. And similarly to karyotyping, any abnormality in this labeling pattern would indicate the presence of a chromosomal alteration. So here there's no culturing step, there's no DNA amplification needed, no denaturation or no fish probe required. And this is how it works. Electrophoretic cycles are applied to the DNA that will be linearized and elongated in a hundred of thousands of nanochannels in parallel, allowing the acquisition of a massive amount of data per sample. This is what you have on the left part of the screen. And by running the instrument a bit longer, you can increase the yield to coverage levels that are simply out of reach of any long-read sequencing technology, and this at no extra cost. On the right panel, you can see the raw data generated with the DNA counterstain in blue and the labels applied by our DLE enzyme represented in green. And one other major difference with long-read sequencing is the size of the molecule that we image that are above 200 kb. So we combine all you need to address complex cancer samples, long-read information at high coverage, and for just a few $100s per sample. Very briefly, the workflow is very simple. You extract DNA with our kit to obtain ultra-high molecular weight DNA of molecules of several hundred kb long. This DNA is then labeled with our DLE1 enzyme and the counterstain. This is where this specific labeling pattern is applied genome-wide. The stained DNA is then simply loaded on the chip where the nanochannels are engraved, and from there, everything is automatic. The DNA molecules are imaged, aligned together just based on the labeling pattern to create a consensus map that we call optical map. We also measure the coverage genome-wide to detect any large gain or loss of genetic material. And ultimately, the sample map is compared to reference. This is what you have at the bottom of the screen. And this reference can be adjacent tissue. This can be a control cell line or simply a reference like HG19 or HG38. And it is this step of map comparison that will identify any deviation in the labeling pattern that would indicate the presence of a structural variation. This is how we represent them with our map alignments. You do have examples of deletion and insertion. So we always represent the reference in green and the sample map in blue. And we will look for a difference in the labeling pattern and the distance between the labels, between the reference and the sample map. So you can see here deletion and insertion, where this spacing between the labels increases in the case of an insertion and decreases in the case of a deletion compared to reference. We can detect the same way repeat array expansion. We can detect this pattern to be duplicated in our sample map, indicating of a duplication. Translocation, when the sample map will align to two different genomic locations. Inversion, of course. And again, we can check genome-wide the coverage to detect any large gain or losses through our CNV pipeline. You can visualize your genome in our browser called Binano Access, and you can see here a circles plot with the cytobands for each chromosome on the outside. Each dots here will represent an event such as insertion, deletion, inversion, duplication. Copy number variation at the center and translocation are these lines connecting these different chromosomes. Every time you would click on one event, this would open the map, giving you a much higher resolution of the event. We will see a few examples. This performance of optical mapping to detect variants that are missed by NGS, and specifically long-read sequencing, can be observed in results from a recent publication from the Human Genome SV Consortium in a well-characterized cell line, not allele rearranged as you would have in leukemia sample. The sequencing-based methods of based on PacBio HiFi missed about 1/4 of the variants identified by Binano, and many of them were overlapping with complex regions of the genome that are known to cause macrodeletion and macroduplication syndrome, for example. We're seeing adoption all over the world, and numerous studies are today evaluating optical mapping against the standard of care for cancer patients to modernize and complement actual workflow. I would develop only on one study and a couple cases that really nicely illustrate how optical mapping can modernize this cytogenetics to identify clinically relevant variants missed by the standard of care and push the boundaries of precision medicine and our understanding of cancer. The first study is a multicentric study led by Bryn Levy from Columbia University and James Broach from Penn State University. And it's really a landmark study where optical genome mapping was compared against the standard of care in 100 AML cases that were previously characterized by karyotyping, plus minus FISH and chromosomal microarray. Binano not only identified all the clinically relevant SVs and CNVs reported by this standard of care. So here you have example of a balanced translocation that were nicely detected here and in comparison to the karyotyping, but also detected in 11% of the cases events that were missed by the routine method. And this is an example here of a cryptic translocation between chromosome 4 and 5 that was missed by karyotyping, while this translocation 821 was the only one identified and reported here. Besides identifying all the clinically relevant SVs and CNVs reported by the standard of care, so optical mapping also refined the genomic structure reported by traditional cytogenomics and identified new variants in about 24% of the cases. And most interestingly, in 6% of the cases of AML, patient presenting a normal karyotype, optical mapping directly identified additional variants, some of them including fusion events that were missed by this method. It's also optical genome mapping identifies way more events than classical cytogenetics because we have a much better resolution and we are not a targeted approach like FISH, for example. But the amount of data generated is not an impediment to an easy characterization of cancer sample. And some SVs, translocations, are considered a hallmark for risk stratification of leukemia patient. And in just a few clicks, you can highlight events that are reported in international guidelines published by WHO or NCCN. And to do this, you just create a panel, virtual panel that we call a bed file that will keep the genome-wide information, but extract from it the events impacting loci of interest that you listed in this bed file. And of course, this panel can be customized at will or combined to another. And we would be happy to assist you creating one and having them evolve over time. I would like to switch to another example, and this identification of a missed variant really has a tremendous potential in precision medicine to bring an answer to patients that are negative for all testing methods. And Dr. Gordon Araka from a CHLA in Los Angeles does a genetic testing of a pediatric leukemia patient, and they routinely observe about 15% of ALL and 5% of AML cases where the key genetic driver remains unknown with their current testing method using karyotyping, FISH, and NGS panel. And some of these negative cases were sent to optical genome mapping. And one case really stand out in terms of discovery. And this sample is from a patient that had a very poor response to treatment and developed residual disease requiring a more aggressive treatment afterwards. And a translocation 2.5 was identified by karyotyping at the time of diagnostic, but they did not really know, did not really understand the significance of this rearrangement. And because we have a better resolution, optical mapping found the same alteration, but could locate the breakpoint at a much higher resolution. And interestingly, this breakpoint was found leading to a fusion involving PDGFR beta. And this event was later confirmed by FISH and RNA-seq. And that's very interesting because PDGFR beta is associated with a phi-like allele and linked to very high risk ALL, as well as being associated to a better response to tyrosine kinase inhibitors. So this patient could have been treated accordingly with imatinib if this translocation was better characterized at the time of diagnostic. But the interesting question is, why was this missed? It's because a fusion of PDGFR beta with the gene SPTBN1 has never been reported in ALL before, and thus was absent of their FISH assay and their OncoKids NGS panel. And it's striking to see the discovery made in cancer sample for which for like leukemia, where the standard of care is already focusing on the identification of a structure variant. And that really highlights the potential of applying optical mapping in solid tumors where classical cytogenetic is not applied as it is in leukemia and where structure variation is largely overlooked. And Dr. Ravinder Kohli evaluated optical genome mapping against a SNP array, and that is part of their routine workflow in brain tumors. And among the different samples, including in the pilot study, they observe 100% concordance with the SNP array result that you can see on this example, with the OncoScan result at the bottom and the optical mapping results at the top. But many other SVs and CNVs and many translocations completely unsuspected were found. Many impacted well-known oncogenes or tumor suppressor genes such as BRAF, EGFR, or CDKN2A. And these results really confirm how structure variants are overlooked in solid tumor and what critical information is provided with a tool such as optical genome mapping. And addressing that complexity without the burden of a tedious data analysis opens new avenues to develop new tools to address difficult questions. One of them is a sample stratification, clustering groups of tumors based on a structural signature. The circuits plots that are automatically generated allow you to do that. And in head and neck cancer, HPV insertion can cause extensive chromosomal rearrangement as well. And optical mapping clusters patients into two groups: those with an episomal form of the virus that will display very minimal levels of rearrangements, from those with an integrated form of HPV showing much higher level of instability. So from this, I would like to conclude and finish saying that optical mapping is long-read technology that really overcomes NGS limitation thanks to its 1600x coverage that can interrogate heterogeneous tissues and allow detection of SVs down to 1% allele fraction automatically and genome-wide for just a few $100s per sample. And conversely to RNA sequencing, it can detect fusions automatically and provide additional information on the mechanisms leading to these fusion events. And from the different evaluation studies that were run, shown the potential and the possibility to identify classical rearrangements, identify actionable and druggable events that would be missed by conventional workflow, but also reconstruct complex structural signatures automatically with that complex bioinformatic pipeline to run. So on this, I would like to thank you again for your attention, and please forward me any question at this email address. Thank you again.